These are tiny molecular machines, and they are doing this inside your body right now. To understand why we have to zoom out every day in an adult human body, 50 to 70 billions of your cells die. Either they are stressed or damaged, or just old, but this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells right now, billions of your cells are dividing, essentially creating new cells, and that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start. The double helix molecules we always talk about this, and this is a scientifically accurate deposition of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has sugar phosphate backbone connected to the sequence of the nucleic acid base pairs known by the letter A, T, G, and C. Now the strands run in opposite direction, which is important when you go to copy DNA. Copying DNA is one of the first steps in the cell division. Here the two strands of DNA are being unwound and separated by the tiny blue molecule machine called a helicase. Literally spins as fast as a jet engine. The strand of DNA on the right has its complementary standard assembly continuously, but the other strand is more complicated because it's run in the opposite direction, so it must be looped out with its complementary strand assembly in the reverse assembly. In reverse, section by section, at the end of this process, you have two identical DNA molecules, each one a few centimeters long but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it's wrapped around potential called histones, forming a nucleus. On these nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form chromosome, one of the largest molecule structures in the body. You can actually see chromosomes under a microscope. In dividing cells, only then do they take on their characteristics shape, otherwise the DNA is more stew inside the nucleus. The process of dividing a cell takes around an hour in mammals, so the footage is from a time lapse. You can see how a chromosome lines up in the equator of the cell. Now when everything is right, they are pulled apart into the two new daughter cells. Each one contains an identical copy of DNA. Now, simple as this looks, the process is incredibly complicated and requires even more fascinating molecules machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, contains the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers which guide and help align them in the correct position. Of the microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different potential working together to achieve multiple objectives. In fact, it is one of the most sophisticated molecules mechanisms inside your body. The kinetic wrap is central to the successful separation of the chromatids. Is created dynamic connection between the chromosomes and the microtubules. For a reason no one yet able to figure out, the microtubules are constantly being built at one end and deconstructed at the other while the chromosomes is still getting ready. Kinetochore send out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically says this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension, where the tension is just right and the position attached are correct. All the proteins get ready, shown here by the turning green at this point. The stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by the dynein motor that is walking gyre. This is the really what is look like like it has long legs so it can avoid obstacles and step over the kinase. Ins molecules, motors that walk the opposite direction. Personally, I am astounded by these tiny molecules machines, how they are able to routinely and faithfully execute their functions billions of the time over inside your body at the exact instant. I am also amazed by the scientists who were able to work out how this happens in such details that we could create realistic depiction of them like you saw in the animation in the video. But perhaps the most amazing thing is just how much is left to be discovered, like figuring out how exactly the chromatid are pulled to opposite end of the cell. There is still so much that we don't quite know. You know what I find exactly is that in science fiction for decades we have been writing about the tiny nanobots that will be injected into our bloodstream that can heal us, but what this suggests the existence of these tiny streams that can heal us, but what this suggests the existence of these tiny molecules machine inside us. It struggled that there isn't a physical limit that would prevent that, and so I think it's pretty likely that in future we will be able to develop our own tiny molecule machines that will be able to repair our bodies better than they can repair themselves.